Fraxa Research Foundation understands that it is vital to engage people around the world. By bringing together Fragile X organizations for World Day, working together, we are changing the world.
Fragile X Australia has been a family support organisation for over 30 years. A key challenge in Australia is ensuring that health professionals recognise Fragile X early, so that children can receive appropriate treatments as soon as possible.
我们成立的初衷是推动中国脆 X 的临床研究和提高社会的认知。我们期待能与世界各地的同行、机构、团体及医疗单位合作，为推动脆 X 综合征的研究和治疗贡献力量。
Approximately 1 in 5,000 children around the world have Fragile X syndrome, and that makes it close to 4 lakh individuals in India alone. Let's spread awareness on Fragile X and make a difference.
En el año 1998, un grupo de padres fundó la Asociación Síndrome X Frágil de Argentina. Nuestra misión es dar acogida a las familias que acaban de recibir el diagnóstico, transmitiendo nuestra experiencia.
Africa as a continent is totally unaware of Fragile X. We have 250,000 people that carry the pre-mutated gene. We've been trying to raise awareness from Cape to Cairo. It's an uphill battle, but we will succeed.
In 2020, Fragile X Center was established in Georgia. Our team is actively involved in raising awareness about Fragile X syndrome in Georgia and helping parent organizations to be stronger together.
Please welcome Hui Kai, Program Chair, VP and Head of Content, WuXi AppTec, to kick off the online event.
Good morning, good afternoon, and good evening, everyone. Thank you for joining us today, and welcome to the 10th episode of WuXi AppTec Rare Disease Webinar Series, Collaborations that Transform. This event series was launched in 2020, focusing on science and collaborations for patients. In the course of this journey, our passionate speakers have shared amazing stories and insights on a wide variety of rare diseases. Last year, our Fragile X event drew more than 5,000 registrations from industry, academia, the medical community, and the patient communities around the world, bringing inspirations to all of us. Today, on Fragile X Awareness Day, we're delighted to partner once again with Fraxa Research Foundation and many other Fragile X organizations around the world to showcase innovations at the forefront of tackling Fragile X syndrome. Fragile X syndrome is a genetic disorder caused by FMR1 gene mutation. This disease is recognized as the most common genetic cause of autism and causes a range of developmental problems, including learning disabilities and cognitive impairment. Unfortunately, there is no cure. Today, we are very honored to showcase more than 30 innovators, the progress in Fragile X gene therapy, gene reactivation, protein replacement, targeted therapy, and drug repurposing, as well as their collective efforts in finding a cure for this devastating disease. Together, we hope to raise awareness about Fragile X syndrome, to foster new thinking that advances scientific breakthroughs, and to strengthen global connections among us. At WuXi AppTec, we firmly believe that a healthy future is only possible when we all work closely together. Thank you for joining us today. Let's tune in now.
Please welcome Katie Clapp, President and Co-founder, Fraxa Research Foundation.
Welcome. We're so glad you're joining us. I'm Katie Clapp, President and Co-founder of Fraxa Research Foundation. I'm also mother of Andy, who has Fragile X syndrome. 30 years ago, Andy was diagnosed with Fragile X, and that started a journey that has entirely changed our lives. We just couldn't accept the future that Fragile X presented to Andy. And so my husband, Michael Tranfalia, and I and another mom launched FRAXA to fund research to offer hope to our family and hundreds of thousands of other families around the world who live with Fragile X. Today, WuXi AppTec and FRAXA present collaborations that transform Fragile X, innovative approaches to finding a cure. Looking through the roster of presenters, I can hardly believe the powerhouses we have in our corner, all pursuing unique paths to treatment and ultimately a cure for Fragile X. This year has seen burgeoning international collaborations, which are funding brilliant ideas and turning those ideas into actual potential treatments for Andy and his peers. And so today, I'm optimistic about Andy's future. When you've heard what these speakers have to say, I think you'll be optimistic too. They have found so many different independent approaches to solving Fragile X. When just one of them succeeds, think of the lives that will change. So thank you for joining us today.
Fragile X syndrome is the most common inherited form of intellectual disability, as well as the leading monogenic cause of autism. With the discovery of FMR1 gene silencing as the cause of Fragile X, we have seen extensive investigations both in pathophysiological and clinical aspects. Where are we now in exploring effective treatments for Fragile X syndrome? Where could be the next breakthroughs? And what lessons learned that are applicable to other rare diseases?
Happy World Fragile X Day to everyone. I want to thank you all for joining us today to commemorate this very special occasion. Peter, this is really a remarkable time to be a neuroscience researcher, especially in the field of developmental disorders, rare diseases, and particularly Fragile X. You've led the way in studying and comparing different single gene causes of autism. What has you most excited right now?
Well, I think over the last 10 years, we've really defined the genetic landscape that leads to at least the monogenic forms of the neurodevelopmental disorders. And we now know that they cluster around sort of subcategories of gene function. And we know there's anywhere between 200 and 1,000 genes that can lead to neurodevelopmental disorders like autism and intellectual disability. And that arms researchers with the tools to make animal models. And genetics has also gone a long way that we can make any model we want to now. We have human stem cell models, we have rodent models, we have large animal models, and we can really address questions that were never achievable before. If you combine that with the new techniques for being able to understand brain circuits, where we can delete a gene in a particular cell type or in a particular cell region, we can really now start to define the pathophysiology associated with these disorders. And armed with that, we can ask much more fundamental questions about what's going awry in the brain in the absence of a particular gene. So it's a really exciting time to properly define these disorders in terms of how they affect the brain, and also ask questions of when can we ameliorate the pathophysiology. So it's a phenomenal time to be a neuroscientist working in this field.
Liz, this seems to be a very busy time for clinical trials in Fragile X.
Yeah, it's very busy. We have quite a few different industry partners that are looking at different targets. And so we're working hard to figure out what are the best ways to go. And I think in the Fragile X world, we've had some failed trials in the past because we were really the first condition that went toward trying to target treatment of the underlying disorder and sort of fix the brain and fix the actual condition itself rather than just using supportive treatments. And so we learned a lot during those trials about problems that we had with the models that were being used at that time, really 10, 13 years ago even. made a lot of progress on new ways of approaching these trials that hopefully will allow some of the agents that are going forward right now to be more successful.
Well, things certainly have progressed since the early days of Fragile X clinical trials, and I think you deserve a lot of the credit for that, Liz. What would you say is the state of the art for clinical trials methodology in Fragile X? And just to extend that, what can other rare diseases learn from this progress?
Yeah, so I think we've learned that you can't just, take any outcome measure from autism and use it in Fragile X. And we've learned a little bit about what patient populations we can use. And I think one of the things we've learned is that the mouse model in particular is very easy to treat, and we can see improvements in the mouse model easily, but then that's very hard to translate to people due to the variability of the condition in people and the heterogeneity of their background and the heterogeneity of where they live and what they do. And so the variability makes it challenging to use measures that, have a lot of variability themselves. And so we've done a lot of work to find objective measures that are going to look at the Fragile X condition better. Things like cognitive measures, like the NIH Toolbox that we've now spent about 8 years validating for intellectual disability, and particularly for Fragile X. Things like EEG that we can measure, circuit activity or brain responses to sound, for instance, that maybe those are not kinds of things that we can get a drug approved on. But they are the kinds of things that can give us an indication that we have a signal so we know which drugs to move forward. And we don't know that circuits and different neurotransmitter mechanisms and so forth are going to behave exactly the same in the mouse and the human. But if we have ways of moving forward into humans where we have some sort of directly translatable markers, like an EEG finding that's the same in the mouse and the human, and we can show that a drug targets this exactly the same way in a mouse and a human, then that makes that a little more translatable to humans than some of the other mouse behaviors we've been relying on. And also, we are now thinking that, we get data in a model organism, and that's great. We really want to have that data to be able to show that a drug looks promising, but we've changed our thinking that you don't just go into a big trial. You need to do an early phase trial where we look at a lot of objective measures and demonstrate that we can see a signal. And if we can't see a signal in a trial where we've looked at what the drug does in a smaller number of patients with objective measures that have less placebo effect, then we may not want to use patient resources to do a large trial of that agent. And I think the strategy of doing a small trial with kind of intensive use of different measures to prove that there's a signal has actually been fairly successful over the recent years. And I think we'll see more successes with that as we go forward.
It is really fascinating to think that you can use human electrophysiology to actually get a glimpse of what's going on in the brain. That seems like a really powerful tool. What are the specific things that you're looking at when you look at human electrophysiology?
So we're looking at both at EEG and something called ERP, which is event-related potentials. And so in Fragile X, we look at event-related potentials mainly related to sound. And that's because patients with Fragile X have an extreme sensitivity to sound in many cases. And when you actually measure their response to sound in the brain, they have a bigger response than you would see in a control person of the same age. And so we can measure this excessive response to sound. There are a lot of characteristics about sound processing surrounding that we can measure. And then there's an EEG background. There's something called gamma power, which is just a certain frequency of EEG activity that is increased in Fragile X. And so we've been able to see in the mouse model that the gamma power is abnormal, just like in the people. And we've been able to see in the mouse model when we use really good detailed EEG that uses multiple leads and not just, a couple leads on the mouse head. When we really look at the mouse EEG, we can see the same kinds of abnormal processing of sound in the mouse model as well. So if we have an agent that changes those things in the mouse, then we can expect, and then we can immediately go and check and see, does that agent change the same thing in a person? And then what we really need to do, so this is work that we don't quite have done yet. What we really need to do is be able to hook up those EEG changes to clinical phenotypes and show that, like, when you change gamma power, you get less anxiety or something like that. Or when you change novel sound processing, you get better language. But we aren't there yet. And there's ongoing work in that direction that hopefully will help us in the future with seeing these EEG changes and knowing then what should we measure clinically that will be clinically meaningful.
So there's still a need to validate some of these electrophysiological methods and to see if they correlate with clinical improvement. But is there a sense that some of these electrophysiologic tests will actually normalize with treatment? Have we seen responses to drug treatment that actually involve changes in electrophysiology?
Yeah, I mean, there are several examples of that. Craig Erickson is running an NIH-funded project where they're looking at changes after a single dose of different agents, and they have seen that some of the agents that they have used have changed the electrophysiology. And we ran a trial of a phosphodiesterase inhibitor, a PDE4D inhibitor, recently a phase 2 study, which actually showed that you could change the excessive sound sensitivity in the patients with the drug. And so that's a very strong finding, particularly when in that study it was coupled with a change in cognitive performance on the NIH Toolbox, which is another objective performance-based measure. And the parents reported that the patients were better. So this was a small, intensive, let's look and see if there's a signal kind of study. And this is an example of the kind of study that we need to do. So now we know we have a great early signal. And so hopefully that will predict better results in phase 3 trials than what we've had in the past.
That really does seem clinically relevant, and it really does seem relevant to quality of life as well. So that's very exciting.
Yeah.
Peter, one of the questions that I hear people ask most often is how treatable developmental disorders in general might be later in life, and specifically how treatable Fragile X might be at later stages of life. Now, you've been a leader in this field in studying development in Fragile X and a lot of other monogenic causes of autism. What's your take on this? How reversible is this later in life?
Well, that's the $6 million question. I mean, Adrian Bird really changed the landscape and the work of Huda Zoghbi as well, showing that Rett syndrome, when MECP2, the gene that's altered in Rett syndrome, is re-expressed late in life or knocked down late in life, you can either, in the first case, you can rescue many of the phenotypes associated with in the rodent models. And if you knock it out late in life, you can recapitulate many of the features of Rett syndrome, which argued that there isn't a critical period for MECP2 re-expression or Rett syndrome. Now, whether that's true for numerous other disorders, needs to be tested, and it is being tested for many disorders, but it's one of the central questions we're asking in my lab and in the center that I work in, and other labs around the world are asking. But I think the most likely outcome of all of this, our prediction, if you will, is that certain models will be more or less amenable to gene re-expression late in life, and we don't know. And the key question there, I think, as well, is it's unlikely to rescue everything when you express late in life. There's different critical periods. There isn't a single critical period. So certain features might be more amenable to genetic re-expression. Other features might be less amenable to that, and we won't know until we try. The other key aspect to that, I think, Mike, is that we're talking, these are largely being tested with genetic re-expression. But even if genetic re-expression has a critical period and you can't rescue certain features of Fragile X syndrome, for example, that does not mean they're not amenable to other types of therapies. Pharmaceutical therapies might work on the circuit, which is still amenable to treatment, even though changing the root cause, i.e. the loss of FMRP, is no longer amenable to just re-expressing it. So I think there's lots of different critical periods. Different features will have different critical periods, different treatments will have different critical periods, and that's really important. And that's why we need just a lot more research into all of these different monogenic forms, including Fragile X.
Huge advantage for rare diseases, for monogenic diseases, for a lot of the things that you're studying, the single gene causes of autism. And this is a huge advantage when it comes to drug discovery and development of new therapeutics. It really is amazingly powerful.
Yeah, it is. And we talk about the rare diseases, but if you combine all the different rare diseases together, you end up with a disorder state that is not that rare. It affects well over 10% of the population. So it's crucial that we understand these, and especially how similar they are, and if we can cluster them together for therapeutic purposes.
So what research still needs to be done to understand development and its role in these neurodevelopmental disorders?
Yeah, so what we're trying to do is cluster the different monogenic forms of autism and intellectual disability, not just in terms of commonalities in the biochemical pathways that might be affected, but commonalities of timing of when the brain circuits start to change, and to really identify, to cluster them based on developmental windows. And one of the things we found recently was if we treat early in life just transiently, we can rescue some of the issues in our models of Fragile X syndrome, that we can remove the treatment and we've rescued that particular stage of development. And other people are starting to find the same things for other aspects of Fragile X. So I think that's actually a really key question that's never been thoroughly addressed, is are there windows that if we can treat during those windows, we can get good permanent recovery? And I think that's a really interesting question, again, about critical periods that we really need to understand more deeply.
Very exciting. Liz, I guess I should ask you the same question about what work remains to be done in the clinical trials field, because we've come a long way, but we're not totally done yet, I assume.
Yeah, we have a great deal of work to do in the clinical trials field. I mean, we always lag by, 10 years or so, the basic science people, in figuring out how to translate things to people, because it's just so challenging. But I would say that the work that needs to be done is, I think Peter's idea of critical windows is very important. And we in clinic have always thought that combinations of medications would be needed to really fully fix Fragile X, because the FMRP has so many roles, and it may have different roles at different times in life. And there may be certain things that work when you're young and certain things that work better when you're older, as Peter was mentioning. And there may be things that work together. But in order to have that kind of treatment, we have to get our foot in the door. The reason cancer has so many good treatments and why they can add drugs onto other drugs is that they got drugs approved at the beginning, and you can only test combinations in general after something is approved, although we are doing some new study designs to try to test combinations of medications. And I think it's important to develop that. So that's an area of clinical science that we need to develop. But traditionally, you have to get approvals of one thing, and then you can treat a patient with an additional medication that's investigational. So trial designs for combinations, we need to get, we've done a lot of work on outcome measures, but at this point, we don't yet have the FDA considering them, quote, fit for purpose. And this is work we need to do with the FDA so that we can generate outcome measures that index the phenotypes in Fragile X and index the important things to families in Fragile X well, in addition to giving us good objective performance-based measures in order to move drugs forward. So we have a lot of validation yet to do, even on the things that we've already done work on. And so I really see us as needing to continue to improve our clinical trial science. We've collected in the recent years a fair amount of well-vetted natural history where we can actually see the trajectory of Fragile X, of typical Fragile X with just standard of care treatment. What do patients with Fragile X do over time in terms of their development based on different measures? And we need to have more of that, because if we had a gene therapy, we certainly wouldn't expect all the effects to occur in the context of like a 3-month trial. We'd need to be able to look and see what did that gene therapy do across life, and to be able to understand that, we have to have these very good natural history studies where we understand what does Fragile X actually look at across life on measures that we can use to demonstrate, you know, what is the gene therapy or what is another drug doing. And so that's you know, early in its collection. We have a new project with the CDC where we're going to collect 600 data on 600 patients that will be across the pediatric age span, really from about 5 to 23, which is going to give us very nice data on how does Fragile X evolve. We've already collected some data through my NeuroNext trial and through an earlier CDC project. And so I think we have a lot of building blocks to get to the place where we have this multiple therapy treatment that we really need. And right now we're in the midst of building the building, so to speak. We're just putting more bricks in the wall every year as we get that basic database that feels like cancer already have, that we can use to really get treatments approved and to improve people's lives.
It does seem like the validation of these clinical endpoints is a real catch-22. It's really even kind of a whole series of catch-22s, because you know, you have to correlate one thing with another thing, which correlates with quality of life, but then you have to show that it moves with treatment. But for it to move with treatment, you have to have a treatment that works, and then you have to just repeat this cycle over and over again. So where are we in that whole process? It feels like we're well on the path, but we're still on the path. We're not there yet.
Yeah, we're on the path. It's a challenge, because yes, we have to show that we can do the measure in Fragile X and that it's reproducible, and then that the measures have content validity and the standard things you would do to validate a test. But then we have to show how much change on the measure is clinically meaningful. And to do that, we have to use interviews of families and other kinds of measures where families rate how much improvement they're seeing in the patient and whether that's a clinically meaningful improvement. So we are actually with NIH Toolbox and some of our family-based report measures of things like the biggest problem that they, like, we have them just pick their biggest problem and then rate it on a scale. We're in active discussion with FDA on what we need to do to get those measures fit for purpose. And in the upcoming trials, there will be a lot of work toward providing FDA what they need to make these measures be what we need to get drugs approved. So it's an ongoing effort, and it really involves an interplay between us doing scientific validation on a measure and then going back to the families and asking them, well, if this measure changes this much and it corresponds to sort of this change in what your child can do you consider that a clinically meaningful change? And so the FDA is very focused on that right now, and that's work that is actively going on in the upcoming trials. So we're going to have each sort of 2 or 3 year period, we get more information on how to best move these forward. But yes, it's a challenge, but we're hitting it head on.
Well, here's a question for both of you, each of you. I want you to look into your crystal balls and to tell me where you see us in 5 years or in 10 years. Peter, where are we heading?
Yeah, well, I mean, I think for all the reasons I said earlier on, I think the future's bright. And from a fundamental research point of view, I think we're getting far better with our models of these disorders in order to get real predictive validity. I mean, that's the holy grail. If we can see a particular therapy having a beneficial effect in our animal models, that will be predictive of what's happening, what happens in the clinic. We have to get a lot better and at the research level in making our models predictive. We need to understand our models better. We need to ask the model. We need to use our models in the appropriate way. And I think we're getting far better at that as scientists. So from that point of view, I'm very hopeful. I think we are going to have good predictive validity. We need a drug that we know or a treatment that we know works well in humans to test predictive validity in our animal models. And I'm really excited about the PDE4, that this might give us that opportunity. But yeah, with all of the new techniques we have, with all of the new knowledge we have, I think the future is very bright for coming up with therapies that are predictive in our animal models for the clinic.
And Liz, what is it that excites you about the future 5 years or 10 years?
Yeah, so I think we're turning a corner, and we know now that there are genetically based therapies in development for Fragile X, as well as small molecules. And it may be that these strategies will actually work best together, or different things will work better at different ages. And so we need to proceed ahead with all of that. And so in 5 to 10 years, I mean, I think we're just getting trial designs really under our belt. And I would hope that in that period of time, we have that initial drug approved so we can start adding things on. We're in the midst of doing gene therapy trials. We've got a process figured out for sort of fast fail, where we can look at animal data, move that into humans, and kind of a standardized approach to do a very, a small focused trial, figure out, does the drug work, and move on to a new target if it doesn't really seem to have an impact in humans. So and that's the way we're going to get to those combination therapies that we want. And so I think we have a lot of positivity going right now. I think we're moving into some big, exciting trials, and hopefully those will be more successful than the past. And I would hope that in 5 to 10 years, we've got at least one drug approved, and we're working on getting those add-on therapies, and we're moving actual genetic therapies like gene therapy into the clinic.
Well, it is a really exciting time, and it's a really hopeful time. And there really is this sense that everything is coalescing, that finally all the pieces are coming together. And I just really want to thank both of you for being here, and happy Fragile X Day.
Yeah, happy Fragile X Day.
Happy Fragile X Day to everyone.
The clinical intervention of Fragile X syndrome has largely focused on symptomatic treatment of psychiatric and behavioral problems, yet to eradicating the disease. We need to tackle underlying causes. What are the current approaches targeting the root causes of this inherited disorder? Will they be the game changers we need in the fight against Fragile X?
Jeannie, what sparked your interest in Fragile X technologies?
I've been interested in Fragile X for a very long time. First, as a PhD student at the University of Pennsylvania, where I trained as a physician scientist. And there, my PhD work was directed at understanding this unusual inheritance pattern for Fragile X. And of course, this was back before there were trinucleotide repeats and before FMR1. What really caught my attention as a student was the fact that the syndrome can only be passed through the maternal lineage, which was very odd. And it had all the markings of an imprinted disorder. And in fact, Fragile X is an imprinted disorder, meaning that the parent of origin has an effect on the gene. And this unusual pattern of inheritance and the fact that it's X-linked is actually what led me to X chromosome inactivation, which has been my lab's main focus for the past 20 plus years. And as we've gotten a better understanding of the basic mechanisms behind X inactivation, we've become interested in leveraging that understanding towards treating X-linked diseases. And those diseases would include not only Rett syndrome, which is something we've been studying for 10 years, but also Fragile X syndrome, which brings me back full circle to my PhD work. And so these past 10 years or so, we've been working on 2 technologies to address the symptoms in Fragile X syndrome. and both of these technologies, well, one is more specific for Fragile X, and the other is something that could be applied to a number of different X-linked diseases. And so yes, I've been interested in developing Fragile X therapies for probably 3 decades at this point.
So what is the approach that you're working on, and how does it differ from existing treatments for Fragile X syndrome?
I mentioned that there were 2 technologies. They're actually very different. They're focused on different targets. So the first one is what we called a selective X chromosome reactivation. And I should mention that this is a technology that we envision working in girls with Fragile X syndrome, as well as girls with other X-linked diseases. And I can say a little bit more about that in a few minutes. But the other technology is much more specific for FMR1. So we call it FMR1 reactivation. And this one can be applied, if it's ever successful, can be applied to both boys and girls because it selectively addresses the mutant copy of the causal gene, which is FMR1. And so you asked the question, how do they differ from existing technologies and existing medicines? And well, the reason we're excited about these technologies is because they really target the root cause of Fragile X, which, as you know, is the absence of FMRP, which is the protein product of the gene FMR1. So unlike the currently available treatments, we're not targeting just the symptoms or individual symptoms of Fragile X. We're actually getting at what is responsible for these constellations of symptoms to begin with.
So can you explain what the X reactivation technology actually involves, Jeannie?
Yeah, right, so there's X reactivation. And so this stems from a realization that we and others had, going back many years now, which is that girls with Fragile X syndrome and other X-linked diseases actually have the cure within their own cells. Now that sounds a little bit odd until you realize that, so girls have 2 X chromosomes, whereas boys have 1 X chromosome. And so it happens that in order to equalize the gene expression from 2 versus 1, the girls inactivate one of their X chromosomes in early development, before birth, long before birth. And so what this means is that girls and women in general will carry around an extra X chromosome their entire lives in a dormant state. So it's not utilized at all. We just carry it around like a, extra baggage. But since that chromosome has a lot of good genes, it occurred to us and others that we could potentially utilize, maybe turn back on some of those good genes on the inactive X to treat the disorder that is caused by the bad gene on the expressing X chromosome. And so FMR1, as you know, is on the X chromosome. And girls have 2 copies of it, boys have 1 copy. So we have now discovered a way to unlock the dormant FMR1 gene on the inactive X chromosome. And we do that without getting too technical here. We do that by targeting a non-coding RNA called EXIST with an antisense oligonucleotide. So it's a very short nucleic acid. And that interrupts the ability of EXIST RNA to maintain X chromosome inactivation. So we now have a proof of concept in cells and in animals that we can get this to work and partially restore, not fully restore, but partially restore expression of these genes by a few percent to maybe 20%, which we believe can be therapeutic. And so the other really exciting thing about this is that the treatment would only have to be given 2 or 3 times a year, right? So the families would show up with their children and go to the hospital 2 or 3 times a year to get a booster shot. And hopefully that would last, for another 4 to 6 months. So if this ever became a therapy, and we're hopeful that it could, I think it would be a game changer for families.
So for parents of children who have Fragile X syndrome, what do you think the Fragile X treatment landscape will look like for them and their children in 10 years' time?
Right. So, biotechnology is progressing at a rapid pace. I think that the treatment landscape will continue to evolve at a very rapid pace over the next 5 to 10 years. So as you know, there's a very healthy pipeline of medicines that are already in the clinic, right? And these medicines are important because they treat the different symptoms associated with Fragile X syndrome, you know, the autism, the anxiety, the seizures, if they're present in the girls and the boys, muscle spasm, I mean, a lot of things. I think it's very important to have better and more types of treatments for these symptoms. And so I see a lot of that coming through in the next 5 years or so. Ultimately, though, I think we want to try to get at the root cause, right? Because by treating the absence of FMRP or FMR1, absence of its expression, we will have the best chance of hitting all of these symptoms at once and not having to go to have this situation of being in a polypharmacy where you have to take 4 or 5 different medicines, a day in order to alleviate all of the suffering. So I think if we can project out to 5 or 10 years, 15, whatever years from now, we're going to be looking at different ways to restore FMR1 expression. And so perhaps, we might see something in 5 years because we are hopeful about the X reactivation technology. There's another technology I haven't mentioned, which I think will be equally important. That's the area of gene therapy. Various challenges associated with the AAV delivery, the viral delivery. There are issues with overexpression of the genes which are introduced and how many cells in the brain that you can hit. But I think that in the next 5 to 10 years, we're going to see tremendous progress in that space as well.
Thank you, Jenny.
Thank you very much for your time.
The silencing of the FMR1 gene and consequent lack of FMRP protein is the major contribution to the pathophysiology of Fragile X syndrome. With the advancement in gene therapy, gene reactivation and protein replacement, we may really be able to target the root causes of this disease. We are delighted to showcase several innovators at the front line of these approaches and their progresses.
My lab is an engineering lab, and we work on drug delivery, which involves taking different types of medicines or drugs to the place where they're needed inside of the body. My lab has been working on a type of therapy called messenger RNA or mRNA therapy for over a decade now. And one of the things that really interests us is figuring out how the chemistry of the nanoparticles that we put this RNA inside, how that chemistry affects where they go inside of the body. Because if we can design these nanoparticles to take the mRNA to different types of tissues, we would then have the opportunity to do types of gene therapy in those tissues. So for Fragile X syndrome, we know that there is a problematic protein associated with that disease, FMRP. And we were thinking that if we deliver messenger RNA into these cells, into brain cells, we might be able to correct or make up for the problems in that gene that contribute to the disease and its progression. The major progress we've made is being able to optimize our nanoparticle chemistry to take messenger RNA into many types of cells within the brain, and to be able to do it in a way where we can access almost the entire brain. There are some delivery systems where the locations within the brain are limited, or you can only deliver to certain cell types. So we're excited that we're in a position to really look at the influence of these genes throughout the entire brain in Fragile X patients.
Our approach in the gene therapy work entails the use of a type of molecule called a shuttle vector to transfer the recombinant protein into the brain. The basic strategy is called gene replacement therapy because the viral genome contains the mouse or human DNA sequence encoding FMRP, the protein that is missing in Fragile X syndrome. These experiments employ the mouse and rat models of the disorder and a specific type of virus called adeno-associated virus, or AAV. The recombinant versions of these AAVs used in both preclinical studies and in human clinical trials are both non-replicating and non-pathogenic viruses. We have made substantial progress over the past several years. The data from our published studies demonstrates partial reversal of the Fragile X phenotype in both the mouse and rat models of the disorder. Our current work is devoted towards testing new AAV constructs, that is, new versions of AAV FMRP, testing new drug administration procedures, and exploring new modes of probing AAV FMRP efficacy in the animal models. As one example of the latter, that is, probing the efficacy of the treatment, we are using electroencephalography, or EEG, to record spontaneous electrical activity in the brain. This type of analysis would be directly applicable to a human clinical trial.
The approach we have taken is based on the identification by Dr. Hervé Moine, our collaborator from the IGBMC, of this enzyme DGKK as the most proximal messenger RNA target of FMRP in neurons. The absence of FMRP leads to the absence of DGKK, and the latter mimics the effect of FMR1 knockdown, both in vitro and in vivo. So to tackle this problem, we constructed an AAV that carries an FMRP-independent variant of DGKK, and we administered this vector directly into the brain of FMR1 knockout mice, which are, of course, a well-known animal model of Fragile X syndrome. And when we did this, it led to the correction of all behavioral abnormalities observed in the knockout mice relative to healthy wild-type mice. Also, treatment with this AAV was not associated with any overt toxicity in the mice. These findings were recently published in EMBO Molecular Medicine. And we believe, on the basis of these findings, that gene therapy with DGKK holds considerable promise as a treatment option for Fragile X. And we are currently performing additional non-clinical studies to advance this candidate therapy into the clinic.
In terms of my focus, in terms of Fragile X research, I think about it in two ways. One is both understanding the physiology of how the brain works in Fragile X, but as a physician, I feel as one of my callings is to think about innovative treatments in Fragile X. And in my case, those tend to be in a few different arenas. We've put together a team here of basic scientists, clinical scientists, psychologists, different types of interdisciplinary specialties to really understand what something like a protein replacement therapy in Fragile X would look like, and understanding how to put that together in both the safest and the best way possible. We've began working primarily in animal models to understand how replacing the Fragile X protein in the mouse will affect both behavior and other aspects of the mouse phenotype. You know, when we think about now, when it comes to the application of humans, how do we think about that? You know, one example I'll give is that when you think about Fragile X in nature, you think about different individuals with different amounts of protein depending on their mutation status. And with different amounts of protein, you can have different strengths and weaknesses. And so when we think about Fragile X gene therapy, you know, this is not, to me, considered a curative therapy, but rather one where would adding protein to an existing system help augment and help enhance some behaviors that could make it primarily something that would really shift the needle in terms of quality of life or cognition, emotional regulation. So those are, I think, some very exciting avenues as we think about the modern era of potential protein replacement in Fragile X.
Over the past 2 decades, our scientists and clinicians have learned tremendously from the clinical development of targeted therapy to change the course of Fragile X syndrome. What have our past experiences taught us? What are still the main obstacles? How can we fully deliver the potential of targeted therapy?
Craig, I was reflecting this morning and it occurred to me that it's now the 20th anniversary of the paper that Kim Huber and I published, suggesting the outlandish possibility that there might be the potential for a small molecule therapy to change the course of Fragile X syndrome. And it's been quite a ride the last 20 years. A lot of high hopes, and some of them have been dashed, but I feel like we're actually in a really good place to, accomplish our mission sooner rather than later.
Yeah, I mean, I really enjoyed joining that ride where I came from the autism field where, no single gene disorders, no clear leads, massive heterogeneity, and moving into Fragile X with the concept of being able to conduct animal studies, to do bench lab studies, and then truly translate those findings into human work. And we've learned a heck of a lot in the last 2 decades. And, in my mind, we think a lot about failed trials, but every quote failure is a lesson learned. And, overall, our lab and our human work is really focused now on Fragile X is one condition, but how do we define variation molecularly, electrophysiologically, functionally, and then match individuals with treatment?
Yeah, I completely agree with you. When we started, there's very little experience in running clinical trials, and certainly not to the standard that would be required by the FDA. And so many unknowns that occur with any drug development program, like what's the right target? What's the right compound? What's the right dose? Are there going to be dose-limiting side effects that we have to take into consideration? Who are the correct patients? What are the right endpoints to measure? But I completely agree with what you're saying, is that every time we do a trial, we learn something new, and we apply that knowledge. So it's very iterative. And I actually share your optimism that we have learned enough now to actually deliver in the near term.
So in our lab and other labs, high density electrophysiology or brain electrical activity, and what are small molecules doing early in phase 1 and phase 2 studies to see target engagement. And we've really moved away from a reliance on pen and paper measures. Now, we think that's huge and will allow us to truly link preclinical to clinical outcomes. But, even that has taken a lot of time. And again, hindsight is 20/20, and I don't think we don't view it as setbacks, but it's really a new generation and a new iteration of trial approaches that are more tightly linked to animal experiments.
Yeah, I hear you. I mean, I think that there's a lot of flying blind, to use another flight metaphor, but if you don't know, you don't have a good biomarker of target engagement, then it's a lot of guesswork to know, like, what's the right dose? Does the therapeutic effect persist, or is there a development of acquired treatment resistance, which it turns out to be a potential problem? And that knowledge would give you so much confidence to say, for example, in the case of your work, there's an electrophysiological signature of altered function. And this signature can be modified by a treatment, and you can learn what is the correct dose to move that biomarker. From the beginning, Fragile X could be viewed, at least some aspects, as a metabolic brain disease, to the extent that it's a dysregulated protein synthesis in neurons. And of course, we now understand that that's a process, a pathophysiological process that contributes to some symptoms. There are other pathophysiological processes like altered ion channel function, particularly potassium channel function, and still other features that are due to simply altered brain development. The wiring diagram is different. And so we would not expect one treatment to hit all of those three causes of dysfunction in Fragile X. And having some insight from a biomarker on, what symptoms line up best with what treatment strategies would be super helpful. Because, you could have a fantastic treatment, but you're measuring the wrong thing. And I think that has been some of the clinical experience, which is very frustrating to the clinicians and the families.
And not to mention layering development into that, where we think about treating, from newborns to toddlers to adolescents to adults, and, where your treatment windows there are. So, when we hear of positive leads in preclinical, data, and we take that into human studies, I think, we've definitely sharpened our tools in quantitative ways, but we also have to look at developmental windows and cohorts maybe being different. And the idea that if we have to study something in adults first and we do signal finding, then we're systematically moving through the ages. We're thinking about males and females differently, so they're both represented. We want to represent everyone, but if we don't control our studies to be able to power and think about subgroups with signal detection, I think we're doomed to fail.
Yeah, that is a really good point also. Then I'm also amazed actually by the work that came out of Peter Kine's lab in Edinburgh, where they found that timely treatment during development could lead to an improvement that persisted for many, many months in the absence of continual treatment. Like it was intervening at the right moment in development could alter the course of brain development in a very effective way to help a long-term outcome. So, I really agree with you, Craig, that we have learned so much, and I feel that we really are on the threshold of being able to apply this knowledge to develop some medicines that are going to help.
I feel like we're finally at the honest point where we interface with treatment developers, around the world, where we can take a small molecule, combination of small molecules in our hands, use all the tools that we've developed to detect signal in a meaningful way. And what's really surprised me in the last 5 years through our work with single dose challenge studies, phase 1A designs with 10 to 24, patients, that it doesn't take huge samples to detect signal when you're using electrophysiology, when you're using computer-based testing, which I think is really great for anyone that thinks about drug development in this field, because your ability to make go, no-go decisions using quantitative measures and then de-risk your phase 2 and phase 3 programs once you have a sense of who the responders are quantitatively really just opens the field up in ways that we've not had before, where companies and other developers faced funding large phase 2 programs with heterogeneous samples, males and females, mosaics, non-mosaics. We're just not in that place anymore because of our tool development, because what we've learned and about how we've improved our linkages to preclinical studies. I mean, a lot of folks now can do preclinical FMR1 knockout mouse electrophysiologic studies, see what their compounds are doing in the mouse, then immediately move into a 1B target engagement study in humans, see if there's synchrony and see which humans are responding, even with small subgroups, because we don't have placebo response with those measures in different ways. So we're very excited about that. And, despite all the quote failures, we're more excited in our lab than I've ever been in my career, because these tools are really poised for success because of, as you said, Mark, everything we've learned from the past.
Yeah, it's been really gratifying to see that sort of these small new clinical candidates keep being introduced. And I think your point is also very well taken, which is that we, with a rare disease like Fragile X, we don't have the luxury of endless attempts to succeed, because clinical trials are so expensive that it's not like a huge indication like major depressive disorder or schizophrenia, where you keep your Alzheimer's disease, where you just keep throwing things at the wall in the hopes that something sticks. We don't have that luxury in Fragile X. So the more informed we can be going into doing the study, the more likely we are going to be to succeed. So thank you for advancing the field in that way, Craig.
Yeah, and I think it's important for us as, human and translational scientists, we use what we learn from, example, our NIH studies to inform our treatment development.
Another feature of Fragile X that always impressed me from the beginning was the evolutionary conservation of some of the pathophysiological mechanisms, literally from fruit flies through mice and rats and humans. And so there is I'm very bullish. I mean, perhaps because I'm a basic scientist at heart, but I'm a firm believer in the relevance of the animal models, particularly in a genetically defined disease such as Fragile X.
And we think about who with Fragile X is best modeled by the available preclinical model. So if it's a knockout mouse, who is best modeled? When we do human high density EEG and we look at an individual signal, is that a signal that matches the electrophysiologic pathophysiology we see in the mouse or not? Is that someone who is going to be best modeled? That's how we think about it now. There's still great utility, but obviously it's not an exact replica of the human experience across the board.
Yeah, I think that's cool. And then the question will become, what is the best behavioral correlate or maladaptive behavioral correlate of the electrophysiological signature? Because then that gets you closer to the quality of life objective. But I would say also on the human side of saying you have an altered EEG signature, what major behavioral domain does that correlate with? Is it irritability, aggression, and so on and so forth? And that's another, objective or challenge that we have.
Not to mention thinking about anxiety, which we all believe is a core lifelong feature, but very difficult to measure. And we're putting a lot of effort in collaboratively across human labs to think about anxiety more objectively and more consistently, because we get regulatory feedback of this is a developmental disorder. People can't self-report anxiety. So how do you measure it? But it's so important to the phenotype. So that's something we really have to sharpen tools in that area to have a chance to think of it as a target, because we all know clinically that if you significantly reduce anxiety, quality of life and function comes with it. But if we can't define it in a logical, consistent manner, how do we use it?
Yeah, I think we still have obstacles to clear to go from where we are today to getting a medicine approved. And of course, one of those is the continual need to educate the FDA and the regulatory authorities on the urgent need and the very fact that it won't be one size fits all.
In my last 15 years interfacing with the FDA, I think they're better positioned now to understand variation in Fragile X, understanding these quantitative measures we discussed. Not that they're going to be primary approvable outcomes, but they can have utility in defining subgroups and understanding at a regulatory level that, yes, you can have an indication for some individuals with a single gene disorder, and that's okay if you're defining them appropriately. So I think that backdrop is going to be key to enabling everything we've discussed.
Well, let's hope that we can close the deal in the near future.
I think we're getting close. This was super enjoyable, Mark. Really enjoyed the discussion.
There has been growing excitement in the Fragile X research community about new developments in targeted treatments for Fragile X. Let's hear from our leading experts about some productive approaches. What are the innovative targets and pathways that these researchers are going after? How can Fragile X patients potentially benefit from their progress?
Zynerba Pharmaceuticals is developing ZYN002, a pharmaceutically produced, non-plant-derived cannabidiol transdermal gel for the treatment of behavioral symptoms in Fragile X syndrome. We believe ZYN002 is unique in that it may reverse a dysfunction of the endocannabinoid system found in individuals with Fragile X syndrome. That dysfunction may lead to the loss of synaptic plasticity and the emotional responsivity that's seen in kids with Fragile X syndrome. ZIN-002 is also unique in that it's absorbed through the skin, and it may be a good alternative for children who cannot take oral medications. ZIN-002 may also cause less GI side effects or gastrointestinal side effects, and it also may avoid some issues with liver function as it doesn't get metabolized through first-pass metabolism in the liver. In regards to our progress with ZIN-002, we've completed our first trial, ConnectFX, in Fragile X, and found that ZIN-002 was well tolerated, and it provided a clinically meaningful change and improvement in social avoidance in children with Fragile X, and those children specifically with at least 90% methylation of their FMR1 gene. Our next milestone is to complete our ongoing confirmatory phase 3 trial called ReConnect, and then file for regulatory approval if the results are positive.
Our approach is really a small molecule development. So this is a novel chemical entity. This is a small molecule. We call it NLX-101. And it's a compound which had already undergone extensive investigation for properties which are antidepressant properties and pro-cognitive activity. So this small molecule targets the serotonin system. Now, serotonin is a neurotransmitter in the brain which is known to be involved in a multiplicity of different brain functions and also in autism disorders. And serotonin is known to be involved in regulation of early development. So the serotonin system is an important neurotransmitter, which is relevant to autism and to Fragile X. But targeting the serotonin system has been a complex challenge for many years because there's many receptors, because it's a very extensive system. And the novelty that we are bringing is that this drug candidate, NLX-101, is different because it really is exceptionally selective for one particular receptor of the serotonin system. And we think that's a very important feature of this drug candidate.
When we study the publications or the current treatment regimen for Fragile X patients, we notice that existing treatment options or drug candidates, they target limited, I would say, some of the symptoms. So we discovered that our candidate, the name is GXB01. This compound preclinically, it has a triple actions on the axis of hormone and neurotransmitter and the neurotrophic factors. And in fact, we also found this compound quite efficacious in addressing by anxiety, autistic behavior symptoms, social stress in non-rodent and rodent animal models. So with this data, and we want to eventually aim to develop a drug that has impact on broad spectrum of symptoms in Fragile X syndrome, and thereby to provide the patients with better treatment options and hope to avoid polypharmacy. We are very happy to announce that it's just June 2022, last month, we received approval for phase 1 trial in Australia, and we have started the enrollment of healthy volunteers for the study. Hope to have the first dose in human in July or the third quarter this year. Then the next milestone will be, of course, safety, the first the priority. So we want to first demonstrate the safety, tolerability in human, and also to prove that this compound has a favorable PK profile in human. We also did some, we want to obtain biomarker data, which will help us to define the starting dose in phase 2 study, at which pharmacological effect can be effected in the patient population.
It's unique, in my opinion, to have a drug, which is Briastatin, and other of the compound drugs that we have which are in our platform. We have many of these. Briastatin is the lead. We think it's fairly unique to have a drug that is specifically targeting synaptic growth and restoration, as well as preventing neuronal death. It also does other things, but its major focus is synaptic growth. The beauty of the Briastatin, which targets something called PKC epsilon, and then its major target, brain-derived neurotrophic factor, BDNF. It also targets other growth factors, a whole repertoire of synaptic growth factors. There's a beautiful molecular pathway that actually begins with gene expression and mRNA longevity that is targeted by Briastatin PKC epsilon. And we've worked that out. And it turns out that, again, it has this essentially diffuse, but very, very focused, diffuse effect on synaptic growth, as well as neuronal preservation.
We're very much an early stage drug discovery project in this case. So as I mentioned, based on the understanding of the disease mechanism and how nerve cells connect with each other, and the synapse is a key part of how nerve cells connect with each other. We understand, based on some basic research, what might be happening at the synapse in the Fragile X syndrome, and therefore we have a hypothesis of how we might be able to have small molecule drugs that can alter the function of a key protein that is involved in the structure and function of the synapse. So we've been funded for this project from the Medical Research Council, so that's the UK government. And what's encouraging for the Fragile X community is that the Medical Research Council clearly recognised the unmet need in Fragile X syndrome, and I guess liked our hypothesis and funded the project. It's been a 2-year project, which we're just coming to the end of. Now we're funded to identify some small molecules that alter the function of the protein that we're interested in. That we've identified those compounds in the test tube, if you will. So those small molecule inhibitors of the key kinase that's involved in the synapse, those compounds are selective, so they affect just this kinase that we're interested in and not others. They work in cellular models, so they can enter the cell and inhibit the kinase in the cell.
The main issues I am trying to address in Fragile X is the brain-related phenotypes. So Fragile X leave us with a very interesting scenario, right? In terms of how we can tackle the brain-related alterations. So hyperactivity, social alterations, cognitive disturbances are mainly the fields I am addressing. And I am very keen of trying to understand how and why this alteration in the FMRP, also in the Fragile X protein, are leading to these alterations, and how these can be then tackled if we really get insight into the mechanisms that are behind those kind of findings, right? So basically what we are keen of doing is trying to help people with Fragile X to overcome all these brain-related phenotypes. Our lab is mainly, we think that we need to really understand what are the mechanisms behind the phenotypes, the concrete phenotypes, because not all of the phenotypes that are present in Fragile X syndrome are driven by the same mechanisms, right? So what we need to do is we need to dissect these phenotypes and then try to understand the mechanisms behind. And then based on that, we can address these possible problems by using something that is different, that is mechanism-based therapies. So we found one of the pathways altered. Well, let's go for it and think about one thing that is very important. That is, Fragile X is not only in brain, it's a multisystemic disorder. So many things that happen in the periphery also affect the brain. And this is something that has only been tangentially taken into account. So our approach are very kind of holistic, trying to also not only focus on drugs, but on multimodal interventions.
At STALICLA, our initial focus in neurodevelopmental disorders was on idiopathic autism spectrum disorder, and our vision was to find new ways to characterize patients beyond behavioral characterization, to be able to translate this characterization into biological and phenotypical markers that could serve as clinical inclusion criteria in clinical trials and result in greater signal at the biological level, and hopefully translate into a better match with therapeutics. And while working in the idiopathic autism field and entering clinical development for our precision autism pipelines, we realized that in Fragile X, despite the condition being a monogenetic condition, there was also a heterogeneity in clinical response to drugs. And so we realized that recent work had established that variation in methylation status in patients could possibly be an avenue to pursue, but that beyond these Fragile X specific variations, the rest of the gene expression of these patients could provide insight into drug response. And we realized that our platform could be applied beyond idiopathic autism stratification to monogenetic disorder stratification. So we engaged in observational biosampling studies to be able to seek first validation of our technology applied in the monogenetic disorder context. And we did this in collaboration with academic centers, revisiting previously failed Fragile X trials to be able to see if we could blindly enrich a population for responders.
Anavex has a strong preclinical and clinical rationale for Fragile X. We have so far a very broad and upstream mode of action that is known to be implicated in neurodevelopmental diseases. By activating the sigma-1 receptor, which confers neuroprotective effects in several models of neurodevelopmental, but also neurodegenerative diseases, including Fragile X. Administration of Anavex 2-73 results in improvement in all these pathologies, and these improvements are overlapping because we believe they are connected caused by cellular stress. And in Fragile X, the genetic dysfunction is causing cellular stress. Another advantage is that Anavex 2-73 is given orally and it's easy to administer. Anavex 2-73 has been shown to be safe. We have developed and seen in our animal models very strong response to BDNF restoration and also restoration of AKT biomarkers, which we're also able to identify in blood. So what we're now doing as a next step is to test the drug, Anavex 2-73, in a Fragile X clinical trial in patients. And we expect to start this year. And we are very excited about it, given the rationale of the ability to restore functionality in these several animal models of Fragile X.
At Kairos, we are developing targeted therapeutics for underlying ion channel dysfunction in Fragile X, ion channel function that contributes to hyperexcitability of the nervous system. And we are specifically focused on a very unique potassium channel known as BK. So I think what really differentiates what we're doing at KRIS in our program on BK from other programs in the space is we have identified a very exciting biomarker that we think is linked to our channel, is abnormal in Fragile X patient, and involves visual function. So the channel is expressed in the eye, and we know that abnormalities in visual function are a key part of Fragile X. We've actually identified using electroretinography, or ERG, which is simply measuring the electrical response of the eye to a flash of light. It can be done very non-invasively in patients. We've actually observed abnormalities in the ERG in the genetic mouse model of Fragile X, and we've actually also demonstrated this in Fragile X patients, studies that we have published in the literature. And what makes that really unique is it gives us a chance to go into clinical trials with a marker that tells us that we're getting our drug to the target we are expecting to hit. And we can move that marker with our compound, so the ability to use that for dose selection and even patient selection as we head into phase 2 studies is really very tangible for us.
So we are developing an oral treatment for Fragile X syndrome, which is an advantage, I guess, although there have been other oral therapies for Fragile X syndrome that have been tested in the clinic. I mean, we have the modality that we're utilizing by addressing a key part of the etiology of the disease has the potential to be disease modifying. And our approach by using early biomarkers such as DEEG, then that can give us greater confidence that the drug is actually reaching the target, and it's actually doing what it's supposed to be doing, for example, by normalizing the neuronal hyperexcitability, which you can see in the EEG. The EEG would allow us, the biomarker EEG work would allow us to establish really early on a PK/PD correlation, which is so important in drug development, but it's relatively challenging in CNS indications, which is the case of Fragile X syndrome. So I think by utilizing our approach by the target that we selected that's central to Fragile X syndrome, as well as the biomarker approach, we can have a very strong way to advance this therapy.
Ours is a novel targeted therapy. We are inhibiting the activity of a protein kinase called S6K1 using a small molecule compound called SOL784. Clown and others have shown that in Fragile X patients and model mice, S6K1 is overactivated in the brain compared to healthy counterparts. And this drives excessive protein synthesis, leading to pathology. To date, the drugs available to Fragile X patients treat symptoms only. Probably the greatest value of our approach is that we have the potential to treat the underlying root cause of the disease. This in turn has the potential to improve profound aspects such as cognition. To date, there has never been an S6K1 inhibitor in the clinic for Fragile X syndrome, so our approach is differentiated over what has come before. So our candidate drug SOL784 has shown promising preclinical effects in several models of Fragile X syndrome, giving us good confidence in taking this molecule forward. We have plans to put SOL784 into phase 1 clinical trial.
As data and new technologies allow for more rapid identification of promising repurposing candidates, many available drugs have been repurposed as potential treatments for neurodevelopmental disorders. In the case of Fragile X, how can we harness the power of data, artificial intelligence, machine learning, and other computational biology approaches to discover new therapeutic uses for approved or investigational drugs? In this conversation, our distinguished speakers will share their insights about drug repurposing.
I'm excited to be here today and learn more about what the companies are doing. And Bruce, with the technical background you have and scientific background, really looking to dialogue with you and learn.
Great. Well, I'm really excited to be here. I think it's important that the Fragile X community learn as much about what kinds of things are happening out in the therapeutic space. And so anything we can do to share information about drug repurposing, drug redevelopment, drug repositioning in our conversation today is really important.
No, that's great. Thanks. I mentioned this to you before. My son has Fragile X, he's 9. And so for me, you know, the appeal here are kind of options, like what's out there, what may be out there, what's on the horizon. And in some cases, you know, what can I do to help? Be part of the trial, cheer on the sidelines, you know, just how to help the movement, how to help kind of move things forward in any capacity. I think as parents and as family members, we're very interested in that.
And as we discussed, I have my nephew, Alex, is now over 40 years old, and he also has Fragile X. So I've been part of his life and watched the progression of his growth. He's been on a couple of clinical trials earlier on. And so it's both personally important to me and scientifically important to me that we advance some therapies for Fragile X patients as quickly as possible. So, you know, I think, Andres, it's important to talk about when you think about developing a therapy for Fragile X. There's gene therapies where we might try and, you know, change things at the very root of the problem in the X chromosome, all the way to things like small molecules that might hit one or two of the symptoms that patients are presenting with. And it's really important to think about that in the way that therapies are developed. And one of the ways that some companies, including the company that I work for, Helix, is developing therapies is starting with known molecules that are human safe. So those could be drugs that have been approved in one or more jurisdictions. Those could be nutraceuticals, which are, you know, the things that we buy at the General Nutrition Center or someplace else that we take, you know, vitamins, minerals, supplements, those kinds of nutraceuticals, almost all of which have strong biochemical impact in the body. They're just not approved as drugs, but they're very useful as potential therapeutics. And then there are molecules that are drug-like, that are in company pipelines, that have either been developed for one thing and maybe it failed, or are being developed for many things that have already been proven human safe in an early phase trial. And so Helix and other companies start with those as a backbone to see, can we find another use for that to treat a disease, a multifocal disease like Fragile X. So, you know, in our case, we've started and came up with lots of different drugs that there's some scientific evidence that there might be an impact in a disease like Fragile X. And, you know, one of the things that companies do is they have different ways of looking at those kinds of drugs or nutraceuticals or compounds to see how they might impact it. One way that we look at is to look at all the scientific information and medical information that's out there and let a computer make connections between each piece of data and every other piece of data that's in our database. And what it lets us do is it lets us find discoverable biology that's already been created through all the research that people have done around all sorts of diseases, including Fragile X, but in lots of other diseases. And the computers, you know, use algorithms that are created by our computational biologists to search through these masses of data and tease out the connections to try and get an evidence chain between starting with a disease like Fragile X and going all the way to some kind of human safe compound that's already available for human use. And so it's really exciting that computational biology allows us to do this kind of thing. It would be almost impossible for a human scientist to keep track of all those data connections and then, get from one end to the other. But the computer can do that and then turn that information over to a human pharmacologist who can then see, does it make sense? Is it strong enough? Is that drug potentially safe in that patient population? What kinds of symptoms is this likely to attack? What might be the potential way that it works, its mechanism of action, its target, the pathway that it's going to interact with. So the computational biology piece added to the starting with a known compound, which some people call drug repurposing or drug repositioning. At Helix, we call it drug redevelopment, because in the end, it seems unlikely that any molecule that's already available exactly the way it is going to be able to, target something as specific as the disease caused by the Fragile X gene issues. But starting with something that seems like it should work and works a little bit, then you can tweak that molecule and make it so that it works better for that patient population. And we think that's a really exciting way to more quickly, more safely get to the patient population, and hopefully with a higher probability of success.
Yeah, I mean, I think for me, I think that's the exciting part about it, is that, the timetable, right, is likely reduced. And so I think about even, again, it comes down to anecdotal, perspective from my own experience with my son, and kind of going through the process to establish what's the right dosing and medication regimen for him that works. And what we found is something that's been effective for him, and it's been modified a bit over the years. And to me, that's almost similar to what we are doing with aggregating this massive amount of data with already, effective and proven drugs, trying to find, maybe it's too much of a simple analogy, but like a cocktail or a recipe that provides a lot of benefit. Because I tell you, where we are now is significantly improved to where we were pre some of these medications when we had them tweaked.
The exciting thing for us is to supplement that kind of, patient-physician general observational research that's going on with the kind of drug redevelopment that would be non-obvious. So, many of the companies like Helix that are working in this space can use the artificial intelligence, machine learning, and other computational biology to find things that wouldn't be that obvious, like, a drug in a totally different class that might actually improve anxiety or improve sleep that hasn't ever been tested for or, used in that context.
Well, I also like the idea of the computer modeling, and the simulations, because it reduces some of the need to get, in a clinic, essentially, right? So when we talk about even clinical study design, just how do you bring somebody into the process with high anxiety and, these challenges that, medical and healthcare treatment will always be necessary because we're humans and we need to care.
Yeah, some of the computer modeling that we'll hope to rely on someday is, can we model what a placebo group would look like so that we don't have to put any living patients in the placebo group and they can all be on treatment? Because I know how hard that is for families that participate in clinical trials, not knowing whether they're getting the treatment drug or the placebo drug.
That's interesting, because I was thinking also about, at the end, at a point where we do have new, effective medicines, well, what's the delivery mechanism? I can tell you, thinking about getting Gael a COVID shot, in itself has been a program. Just how do we work to getting his vaccine shot, which is a challenge. So thinking about a medicine, how do we get him in a future point in time, if there's a medicine, if it's going to be delivered through a syringe, which is going to be a challenge. If it's a pill, we've come up for solutions for that, and luckily we got him to take solid dose pills with a nice dosage of Nutella on the spoon, and it seems to work. So how you administer, especially for someone that has high anxiety, then you, like I said earlier, you kind of create a barrier for other health treatments that are essential as well, something to consider. But, yeah.
Well, I look forward to all the other work that we're going to do together with the Fragile X community. It's really been great talking to you. I wish you and your family, and especially your son, the best. And we'll keep plugging away at Helix and other companies to make sure we can deliver therapeutics that are safe and efficacious as quickly as possible.
Well, thanks for all you do. It was a pleasure talking to you. I learned a ton. And, for all those out there doing awesome work in this space, just thank you. We keep checking in, and we're excited about what's to come. So thank you.
Repurposing of drugs to treat both common and rare diseases is becoming an increasingly attractive proposition to many researchers. Let's learn from our next panel of innovators on their current drug repurposing strategies for Fragile X syndrome. What are they most excited about? What's next milestones they hope to accomplish?
The key about any therapeutic development is having a target, and then having a target that is selective and potent, with a safety profile that would make this tolerable and safe. This is safe and tolerable. It's easy to administer orally or in a liquid formulation. Our approach for Fragile X is really focusing on mechanisms of disease and how a drug may have influence on some of the mechanisms of disease. So we're developing a broad neuroprotective agent. This is called pridopidine. This is the most selective and potent sigma-1 agonist around. And selectivity is very important for optimal efficacy and safety. And this acts to enhance neuroprotective pathways, increases BDNF, has big impact on synaptic plasticity, and would have widespread utility in numerous neurodegenerative and neurodevelopmental disorders. In the past, we've shown that this drug can influence synaptic plasticity, stabilize spines, alter and restore calcium regulation, and also enhance BDNF deficiency, where BDNF deficiency is a feature of Fragile X. It also enhances the transport and development of BDNF secretion. It also has impact on mitochondrial function. It reduces ER stress and enhances autophagy, all of which may be important mechanisms which play a role in the pathogenesis of Fragile X. So we're targeting sigma-1, which has broad effects in terms of secondary effects, and this drug appears to be the most potent, safe, therapeutically tolerable, and is specific for these particular mechanisms. It's a novel approach because it is the most selective sigma-1 agonist. And this would be suitable also in giving in combination therapy with gene therapy or cell therapy. And we've already shown demonstrated efficacy in preclinical models done under the supervision of Dr. Pulati, and also in other neurodevelopmental models, including vanishing white matter disease and neurodegenerative disease like Huntington disease. So the establishment of this particular path of a drug that impacts these pathways in a very selective, safe, and tolerable way is what differentiates our approach.
I believe the greatest differentiated value of our modality is the fact that we're developing models that more closely mimic the human physiology, and so these are cell culture-based systems that are amenable to high throughput screening, that can easily or more easily be used in drug screening studies. They're also models where we can better reproduce some of the complexity of the human brain. And we believe that this allows us to better understand the complexity and the clinical features in patients.
Our approach builds upon decades of progress in genetics and fundamental neurobiology. Mark Baer and I pioneered a new approach to the development of disease-modifying and life-altering treatments for Fragile X syndrome. Specifically, individuals with Fragile X syndrome can be distinguished by a common set of physical characteristics and behaviors, thus defining a syndrome. Once it became possible to perform molecular genetic studies, it was discovered that disruption in a single gene, FMRP, that prevented the expression of a single protein, was the cause of Fragile X syndrome. The next step was scientific discoveries that made it possible to introduce this disruption in this very same gene in animal models. And once we were able to do that, we could understand how the deficiency of this single protein interfered with brain development and developed therapeutics that specifically corrected the molecular impairments that cause Fragile X syndrome. Now, we identified 2 different classes of drugs with 2 different mechanisms of action and brought those into clinical trials. And arbaclofen, our lead product, produced the greatest magnitude of benefit in humans with Fragile X syndrome. And we're currently focused on gaining regulatory approval and making arbaclofen available to individuals with Fragile X syndrome as soon as possible.
The uniqueness of the approach that Curis AI is using is that the drug combination that we've discovered and are using, originally discovered by Professor Nissim Ben-Venisti of the Hebrew University in Israel and in-licensed by Curis. The approach we're taking for Fragile X is really a novel one. We're using a bio-AI platform, meaning a platform that combines AI together with patients on chip and stem cell genomic diversity to get a better simulation of the human body, and in this case of the Fragile X model. The uniqueness of the approach that we are taking is that a drug is offered which addresses the root cause of the disease. The root cause of Fragile X is an epigenetic inhibition of the FMR1 gene, causing a lack of FMRP protein in the brain. The drug combination which we developed prevents the inhibition, the epigenetic inhibition of FMR1 gene, and hence, if successful, would allow a reversal of and the curing of the disease itself. This is not a downstream approach of addressing downstream proteins or genes or symptoms, but rather treating the root cause of the disease.
Purposeful is taking the drug repurposing route. We are looking at small molecules, marketed drugs and nutraceuticals, so agents that have already been tested, and we know their safety profiles, and we're looking for either the drugs or the combinations. I think the main difference from our side is explainable AI, which is significantly different compared with black box methods on the technological level, but also the fact that we do drug repurposing and the fact that we do combinations, we realize that Fragile X is a complicated disorder. We believe that the solution to relieving multiple phenotypes lies in combination therapies. Our approach is to use these combination therapies to better alleviate phenotypes both on the behavioral level and the cognitive level. We suggested a handful of candidates that could possibly work. We validated in the animal model, and now we are in the middle of a pilot phase 2 trial at Rush, which may be completed by the end of the year.
Our approach is quite different from what has traditionally been done within the field of drug discovery in general, but even more specifically in the field of Fragile X, in the sense that what we have seen, and if I focus specifically on the field of Fragile X, we generally see two approaches in the field, which is companies who develop a drug non-specifically for Fragile X and think that there might be some biological reason why it would also work for Fragile X, but they're starting with a drug that has already been developed. The second traditional approach that we see is people doing screenings, so trying every drug that is out there and seeing if one of them work on Fragile X, which is really this repurposing approach. What we're really trying to do using these machine learning techniques is using data from these trials to figure out what biologically drives symptoms of patients and which drug targets can we extract from all of this data that has been generated out there over the many years that there have been research on Fragile X. And can we find drugs, once again, in a computational manner, that selectively modulates these drug targets? So we're trying to do much more from a computational perspective, and we're trying to do it in an agnostic way. So we're not starting from the existing drugs is really like a starting point for us. But we're trying to find something that is really specific to Fragile X to ensure that we have the best possible response in patients.
My research and the way we have approached Fragile X is to look at sort of the deep molecular profiling of synapses. The technology we're using allows us to look at, dozens of proteins within every single synapse of a single piece of brain, right? So this way, what we can actually do is we can use the molecular profiles of each synapse and classify them into individual classes. And then we're able to look at the subtypes specifically in terms of how it changes in Fragile X and how different types of drugs given to the animal actually affects these synapses. And so what we actually found is actually really quite fascinating. I mean, a lot of synapses, if you compare the wild type sibling to the Fragile X sibling, are not changed, but there are very distinct subpopulation synapses in very distinct regions of the brain that changes with the loss of the FMRP protein. So I think what you're actually seeing in this case is that we are talking about a developmental disease, right? There are going to be certain drugs that are going to be good for certain types of synaptic changes and not others. So my technique allows us to really sort of get at the root of these different types of changes and how they respond to pharmacology. And the goal is to really, figure out which mechanisms changes which type of synapses. Can we design synergies with combinatorial treatment to actually get a more complete rescue? And what are those particular synapses that, regardless of what type of drugs, you just can't change? And how meaningful is that in terms of the behavior?
One of the most important works we are doing recently is to derive induced stem cells, iPSCs, from patients with distinct behavior and brain electric activity phenotypes. The reason we want to do that is because there's a really diverse kind of representation and severity among Fragile X patients. And this diversity has contributed to a lot of the complexity in clinical trial design and readout. And some of the clinical trials may actually have run into this complication of data interpretation because of diverse representation of a patient severity and symptoms. So because of that, we decided to derive the iPSC stem cells from the patient with a distinct representation in their severity and the drug response. And this is a collaboration with clinicians and also basic scientists and computation scientists. And our goal is to try to identify, so what are the underlying mechanism for these differences in severity And can we develop an in vitro model in the cell culture model that we can test some of these drugs before they need to be applied to human clinical trial or patient treatment. So this is really exciting because this is highly relevant to what actually need to be applied to patients in the future, and highly relevant to some of the more pressing and fundamental questions in Fragile X research and treatment.
Please welcome Richard Saul, Head of Boston Office and Senior Advisor of Strategic Initiatives, WuXi AppTec, who will close out our event.
Fragile X syndrome is the most common known cause of inherited intellectual disability worldwide. Although there is no cure for Fragile X yet, our distinguished speakers address their vision of the future and the promising technologies that will take us across that journey. Through collaboration, we will provide unwavering support to every family affected by Fragile X. We have a shared vision that advances in sciences and technologies coupled to strong patient advocacy will lead to a significant improvement in the lives of those affected by Fragile X, and perhaps a cure for the disorder. We all look forward to working together in the years ahead to realize that shared commitment, doing the right thing for patients and their families across the globe, and fulfilling the promise of future medicine in a multidisciplinary, collaborative environment. On behalf of WuXi AppTec family, I'd like to express my gratitude to the distinguished speakers for sharing their perspectives and remarkable insights, to the FRAXA Research Foundation and many other supportive partners for your wonderful collaborations in the programming. And to our audience, thank you all for joining and participating from all over the world. At WuXi AppTec, we all believe in a future where every drug can be made and every disease can be treated. This future is only possible if we continue to work closely and collaboratively together. Thanks again for joining us, and we look forward to seeing you at the next episode of our Collaborations That Transform series. Thank you very much.
Thank you.